Thank you, Peter, for the introduction, and thank you to everyone for joining us today as we talk about advances in the field of immuno-oncology and how we're using the technology of flow cytometry to drive those advances. We'll look at specific techniques and new methods that are commonly used in this field to drive advances. So it seems like almost every day we hear about new breakthroughs in cancer treatment. These are called things like immunotherapy or cell therapy, and in the context of cancer, we can refer to these as immuno-oncology. See these, it seems like almost weekly, on the cover of major journals and in the scientific press, as breakthroughs are made, as new indications are approved for immunotherapies, and even increasingly we're seeing this in the popular press, popular magazines and newspapers, bringing really to the public eye how these advances are changing the lives of people with diseases that at one point had a very poor prognosis. So many of these new treatments are proteins, and in many cases they're even cells. So cell-based analysis technologies become crucial for every step from discovery and development through translation and clinical trials. And as we see in a characteristic plot describing the success of a treatment known as a Kaplan-Meier plot, we see the progression-free survival of patients with one treatment compared to patients with another treatment. And as an indication of the dramatic success that's being seen with some of these new treatments, not all of these patients with some treatments ultimately succumb to their disease. We're seeing what can now be called a cure in some cases. And so when we compare across different types of therapeutics for cancer, we can look at a similar model of a Kaplan-Meier survival curve and look at the percent survival with just chemotherapy. And with the advent of targeted therapy, we move this curve to the right, indicating that we buy these patients a little bit more time. What we're seeing in some cases with immunotherapy, whether that is checkpoint therapy or other approaches, is that this curve doesn't reach the bottom in some cases. It actually plateaus, indicating that a subset of these patients have a durable response. And we're seeing an increased focus on combinations of both standard chemotherapy, targeted therapy, and immunotherapy with the expected increased benefit to these patients. And so we'll give a brief introduction to what these different types of therapy are and what they mean and how they work, and then look in a little more detail at how the technology of flow cytometry is really foundational to driving these advances. So immunotherapy is a broad term. In the context of cancer, it encompasses a variety of different approaches, each that has the goal of, in some way or another, restoring our immune function, the function of our immune cells. So there's a wide variety of these, whether they're therapeutic antibodies, checkpoint inhibitors, chimeric antigen receptor T cells, cancer vaccines, cytokines, even some oncolytic viruses. And these are some of the more common varieties. Many more are in development. This field is remarkable for its diversity and the wide range of therapeutic approaches and the really amazing creativity of researchers trying to solve this problem. So for today, we'll focus on top three here, therapeutic antibodies in some various forms, one of which is inhibiting checkpoint blockade, checkpoint inhibitors, and then we'll look at CAR T cells as well. So as Peter mentioned, I'm a research scientist at Thermo Fisher Scientific, which is dedicated to providing tools to support research, including the field of immunotherapy and cancer therapeutics. And we offer solutions across the workflow, whether that is media required to grow your cells, transfection reagents, our OncoMine next-gen sequencing tools, which become critical for deciding which patients can benefit from which therapy. And then today I'll show many examples with our second-generation Attune NXT Flow Cytometer. Now, flow cytometry is a foundational technology for discovery and development in immunotherapy. This is because it allows high-throughput single-cell analysis of a large number of cells. So we can look at millions of cells at the single-cell level with many different simultaneous measurements to get a holistic view of the tumor microenvironment and to look at circulating cells. And so as cells in our sample pass through a laser, the fluorochromes that we attach to those cells are excited at given wavelengths and emit different wavelengths of light, which we can detect on photomultiplier tubes. And as we multiplex many different fluorophores, we can get good indication of not only the phenotype or the identity of our cells, but also measure many different functions of those cells. So many of the examples I provide today will be demonstrated on the Attune NXT Flow Cytometer, including how the capacity to analyze 14 colors simultaneously with multi-parameter immunophenotyping gives us a lot more information on each sample, and how the extremely high flow rate enabled by acoustic focusing lets us analyze samples extremely quickly, but also gives us an unparalleled resistance to clogs, which becomes critical when we're looking at sticky samples like tumor-infiltrating lymphocytes or dissociated tumors. And so fast also in the sense that we can look with automated high-throughput analysis at 96 or 384 samples at a time. So we can move on and look at three different focus areas of cancer immunotherapy and how flow cytometry is driving the discovery and development in those fields. So first, let's look at therapeutic antibodies. Now, many of us are familiar with the structure of an IgG antibody in some aspect, but to summarize, we have 4 polypeptide chains that are both covalently bound together and associated in other fashions. 2 antigen binding fragments, the Fc tail of our antibody, and on the antigen binding fragment, we have a specific antigen binding site. Now, the workflow of someone who is developing antibodies for biotherapeutics covers a wide range and a wide variety of different screens and different assays. Some of the more common things that are done with flow cytometry include a demonstration that our engineered library of antibodies binds to our antigens in the absence of cells, and also in the presence of cells, which can be called receptor occupancy, to show the degree of binding of our antibody of interest to our intended antigen. Also critically, we need to demonstrate not just cell killing or potency, but specific cell killing of our cells of interest. And also, in some cases, we need to demonstrate that our antibodies not only bind, but also internalize into our target cells. So with antibodies as a drug, they use many different mechanisms with which to kill cancer cells. I've heard antibodies referred to as the henchmen of the immune system, because they may direct the potent cells of the immune system to target kill other cells. Their effectiveness as drugs can be changed by modifying them. This can be good and bad, but both pharmaceutical companies and regulatory agencies are very interested in understanding and controlling the exact structure of antibodies, how minor tweaks to their structure have profound changes to their function. So when we're looking at different mechanisms of cell killing via antibodies, this takes many forms, and a few of the key methods here include ADCC, or Antibody-Dependent Cell-Mediated Cytotoxicity, where an antibody is able to recruit killer cell to kill our target tumor cell, or to recruit a macrophage via antibody-mediated phagocytosis, direct induction of cell death following antibody binding, reduction of the complement system to initiate complement-dependent cytotoxicity, and also a unique form of antibody in which a cytotoxic drug is conjugated to form an antibody-drug conjugate. So I'll show you how we analyze and screen for these different parameters using flow cytometry. So first, let's look at ADCC, or Antibody-Dependent Cell-Mediated Cytotoxicity, where in this case, our antibody, our therapeutic antibody, in a in vitro model will be added to our cancer cells of interest, which we'll typically call our target cell. Now, ADCC is mediated by cells displaying Fc gamma receptors, such as Fc gamma receptor 3A expressing NK cells. In this type of experiment, a common approach is to label our target cells, that is our cancer cells, with a dye such as calcine AM, or in other cases with an apoptosis indicator such as the caspase 3,7 substrate, cell event caspase 3,7 green. We can label our target cells with one of those compounds, load our cells into a 96-well plate, and then add varying doses, increasing doses of our antibody, incubate for 20 or 30 minutes, allowing the antibody to bind to antigen on our cancer cell. Then typically either peripheral blood mononuclear cells or isolated natural killer cells or other cell types are added, typically at a high effector to target ratio, sometimes in the range of 25:1 NK cells to cancer cells. Now we put these in an incubator, allow the ADCC to occur for 4 hours, typically gives us a good window process. And then in the case of calcine AM labeling, we expect our natural killer cells to lyse our target cell. In that case, calcine AM will leak out of the cancer cell and have a dim signal, which we can quantify with flow cytometry. In the case of cell event, caspase 3,7 green, we will see an activation of this probe as apoptosis is initiated, giving us a very bright indication of cell death. Where calcine can occasionally be pumped out by membrane pumps in our cancer cells, it may make sense to look at various different models to provide this answer. But after we've done this experiment, we can move on and do a flow cytometry analysis. And in this case, looking at the percent cell death or percent cell lysis indicated here, two different B-cell binding antibodies, rituximab and obinutuzumab. As we increase the dose of these drugs, we have an increased percent of cell lysis, indicating that in the case of obinutuzumab, with Fc glycan engineering, we have far stronger binding to NK cell Fc receptors and more potent cell killing via this mechanism. So we can move on and look at a second mechanism, in this case, antibody-dependent cell-mediated phagocytosis. So in this in vitro model, again, we begin with a similar cancer cell, which again we'll call our target cell. Only this time we'll add a macrophage in addition to our antibody. And in a similar fashion, Fc receptors on our macrophage will bind to the Fc tail of therapeutic antibodies bound to antigen on our cancer cell surface. Now, we can perform a similar workflow. In this case, we can label our target cells, our cancer cells, with either a cell trace dye. A cell trace dye is a dye that covalently binds throughout the cytosol of this cell, permanently labeling it as a target cell, so that in our downstream analysis, we can identify clearly which cell is a target cell and which is our macrophage. Another approach is to label this cancer cell with pH-sensitive dye, such as photo-red or photo-green dyes, which in either of those cases, our cell will be non-fluorescent until it is ingested and phagocytosed by a macrophage, at which point it will become brightly fluorescent. So either of those approaches are very useful to analyze this mechanism. But either way, we can label our target cells with one of those dyes, load them typically into a 96-well plate, and then add varying doses of our antibody. It's commonly done on ice to prevent trafficking or internalization of our antibody, just to allow the antibody to bind to the cell surface. Following a wash step, we will add monocyte-derived macrophages, which have been grown from peripheral blood mononuclear cells from a donor. So over the course of about a week, we'll derive our macrophages, add these to each of our wells at a ratio in the range of 4:1 effector to target cells, incubate long enough for our phagocytosis to occur, and then commonly we've already labeled our target cell with either a cell trace dye or something else. Now we need to identify our macrophage. With flow cytometry, we can label our macrophages and identify them with antibodies against like CD11V and CD14. And then using flow, we will identify cells that are positive for both of these markers, CD11B and CD14, also positive for our target cell dye, indicating that the macrophage has ingested cancer cell. So when we actually visualize the data from this type of experiment, we see somewhat of a dose-response curve showing percent antibody-dependent cellular phagocytosis of various B-cell lines here. And so as we increase the dose of various antibodies, we see that some have a profound effect on the ability of macrophages to recognize and ingest those target cells, and some appear not to have an effect at all. In this case, isotype controls provide a valuable measurement of the lack of antibody-mediated phagocytosis model. So move on and look at one more approach to cell death. And so in this case, we can call this apoptosis, or we can call this the induction of direct cell death via antibody binding. So some of the recently engineered glycoengineered antibodies, especially type 2, will bind to target cells in such a way that just by binding often to two antigens, they will directly induce cell death without the requirement for any effector cell to be present. So we can measure this using the probe I mentioned previously, the cell event caspase indicator. And in this case, we will just load our target cells with a probe of this variety to indicate cell death in a dynamic fashion, where this caspase probe will be non-fluorescent until cell death occurs, until our antibody induces direct cell death. So we can load our target cells with this dye and put them in a 96-well plate. Again, add varying doses of our antibody, incubate for the desired length of time, and then directly quantify apoptotic cells. I mentioned earlier the Attune NXT Cytometer and its high-throughput capabilities. It's enabled by the Autosampler, which provides the same efficiency but with very high sample throughput. And so as you see in this graphic, the Autosampler sits directly next to the Attune, so that we're able to run tubes and plates simultaneously in the same experiment to allow a very easy high-throughput analysis of our samples. And so if we have loaded a 96-well plate with cells and with an apoptosis indicator and our therapeutic antibodies at varying concentrations, we can now incubate those cells and ask the Attune to analyze each of the 96 wells for us automatically. So in this example, we can load 4 different treatments, each in triplicate, at decreasing doses across a plate. And rather than displaying this as a line graph, as we have earlier for all of the examples, the Attune NXT software enables us to directly visualize the results of our experiment without having to export and analyze offline. We can do this simply by toggling to a heat map view, which will give us a quick indication both numerically and visually of the extent of cell death with each of treatments in our cells. So we can see here, both with the color and with the number inside each well, that we have dramatic cell death, a very high percentage of cell death with this treatment, where with other treatments we see little to none. So it provides a very easy view into the results of our experiment. Very quick look at the results of our screen here. So one final mechanism to mention regarding how antibodies are able to act as drugs. And in this case, we'll look at antibody-drug conjugates, which are composed of a therapeutic antibody linked to a cytotoxic drug. Now, the antibody is typically designed to recognize a tumor antigen. They will bind to that tumor antigen and be internalized into the target cell. At that point, a cleavable linker will release a very potent drug into the cell. So these specially targeted delivery vehicles all aim to increase the maximum effective dose that can be delivered to tumor cells. And because the drug is sequestered on an antibody, they can carry new chemotherapeutic agents that would have been far too toxic to deliver via traditional IV therapy as chemotherapeutics. And so when characterizing antibody-drug conjugates, we'll need to know whether our antibody of interest will bind to antigen, as we mentioned earlier, but also whether it will internalize into our target cells, and as we see in the graphic here, deliver the drug to either tubulin cytoskeleton or to DNA of our target cell. So we can measure this very easily by labeling our antibody with a pH-sensitive dye, such as Frodo-red or Frodo-green amine reactive dyes. We can add this labeled antibody to our target cells, incubate at 37 degrees for generally an hour, sometimes as long as overnight, depending on the biology we're interrogating, and then we can directly quantify internalization. In this case, because Frodo dyes are fluorogenic upon internalization, meaning that they are dark when they are outside the cell and will become very brightly fluorescent when the antibody they are attached to is trafficked into lysosomes. And we can see that both visually under a microscope, as in the top left we see Frodo-red conjugated trastuzumab. We only see signal in punctate spots inside each of those cells. And we see that that pattern correlates very nicely with the green spots indicated by lysotracker, and the red signal is very different from the blue circles, which indicate the nuclei of our cells. On the right, we can see that the Frodo-red trastuzumab overlays very nicely with lysotracker, as suggested by the yellow pseudocolor. So visually and qualitatively, we can see that this traffics into the right place. Because this is a very bright fluorogenic signal, we can perform this experiment using flow cytometry also. And so in this example, 4 different breast cancer treatments were performed. 2 negative controls on the top, where we would expect not to see internalization of our target antibody. And then on the bottom left, where our HER2 expressing breast cancer cells were labeled with trastuzumab, labeled with photo-red, we see a very bright population of cells, indicating that this subset of cells has internalized our antibody, and that this antibody is now a good candidate to become an antibody-drug conjugate. And then in the bottom right panel, we can see with the treatment indicated here that we have now knocked out the ability of those cells to internalize that antibody, potentially antibody-drug conjugate. And in this case, explaining one potential route of cancer stem cell immunity to these drugs. So that was a summary of the different ways we use flow cytometry to evaluate the discovery and development of therapeutic antibodies for oncology. So now I'd like to move on and discuss some ways that flow cytometry is used in the discovery and the pursuit of better checkpoint inhibitors. So a new class of drugs called checkpoint inhibitors works by releasing a brake that stops the immune system from attacking tumors. This immunotherapy has been approved for many kinds of cancer and found to extend the lives of patients with advanced disease. Now, the problem is that for many patients, and often most patients, immunotherapy doesn't work. But we see a fantastic benefit to a few patients. So the challenge is really knowing in which patients may benefit. And so there's a great deal of work looking into the tumor microenvironment and also with next-gen sequencing to decide what biomarkers are appropriate to decide which of these targeted therapies are of benefit to which patients. And so when we're discussing immunotherapy, in this case, meaning checkpoint inhibiting antibodies, you'll often hear the term PD-L1, indicating the programmed death ligand 1, which is expressed both by tumor cells and by immune cells that infiltrate into tumors. This can bind to the programmed death 1 receptor on activated T cells. And upon binding, those T cells, which are attempting to invade a tumor and kill it off, are now blocked from doing their job. And so the goal of drugs such as anti-PD-L1 antibodies and many other similar varieties is to mask this brake applied by the tumor cell, enabling our infiltrating lymphocytes to do their job and to kill these cells. And so if we look at a tumor tissue section of a patient's tumor, we can see in the brown stain here on the left column an indication of the tumor parenchyma. The red line on these plots indicates the margin of the tumor in this biopsy sample. In the second column, we see CD8 positive cells, which have recognized that a tumor is present and are attempting to invade and kill it, but they are blocked by checkpoint blockade signals on these cells, either the expression of PD-L1 or other similar antigens. And so what we see is a marked absence of tumor-infiltrating lymphocytes within the tumor, while meanwhile they're gathered around the tumor, poised and ready to attack. And so the goal of the checkpoint blockade antibodies in this example is to block this stop signal provided by both the tumor cells and by other tumor-infiltrating immune cells, enabling the T cells to invade and do their job to kill these cells. And so we can see in a patient where biopsies were taken over time, both before treatment, on the left, where we see almost no brown dots, almost no CD8 positive tumor-infiltrating lymphocytes. But as progress through anti-PD-1 therapy occurs with pembrolizumab, we see after 27 days, we're beginning to have tumor-infiltrating lymphocytes invade this tumor. And after more time progresses on the right, we see a very marked increase in invasion of these lymphocytes, which are now able to not only identify but bind and kill our tumor cells. So in this type of analysis, it's critical to have a very strong understanding of what's going on in this tumor microenvironment. Flow cytometry plays a very valuable part here, where we have many different types of cells to identify, but also we would like to know things about those cells, such as their activation state. So using flow cytometry, in this case on the Attune cytometer, we're able to gate on our tumor cells of interest, our tumor-infiltrating lymphocytes, gate them for viability, remove aggregates, which in the case of Attune, where we are using acoustic focusing, and it's remarkably resistant to clogging. So in the case of a dissociated tumor sample, where our cells can be large and sticky and often very frustrating as they're prone to clog flow cytometers, we don't experience that issue with the Attune. So very, very useful for this workflow to have an analysis where all of our cells are captured. And so in this case, we can look at not only our tumor cells, but infiltrating lymphocytes, and then subcategorize those into natural killer cells, B cells, subsets of T cells, and then look at the activation state of each of these subtypes of cells, where not only the presence of those cells is critical, but their activation state and additional details about those cells is important to decide what route to pursue and whether this patient is a good candidate for these therapies. Now, with samples such as tumor-infiltrating lymphocytes or associated tumor cells, often these are very small samples. They're small biopsies, and we don't have many cells to begin with. And so it's very useful to have as many answers as possible from each sample. So the ability to label with 14 different colors and measure simultaneously that number of different markers gives us just that much more information about each sample. Now, if these samples are very small, we run the risk of very quickly running our entire sample through a flow cytometer. On the Attune, we're able to dilute those samples, take the same number of cells, but dilute it up to a very large volume, and then run it through the instrument slowly at first to observe that everything appears as it should in our analysis. And then once our analysis is set up correctly, we can take that dilute sample of cells, turn the speed on the Attune up to very fast, to 1 milliliter of sample per minute, and very quickly analyze that entire population of cells without the fear or the risk of losing cells from our sample. So in this case, not only the speed, but the ability to look at many different markers is very beneficial for interrogating the impact of immunotherapies on tumor-infiltrating lymphocytes. One final field to mention in the area of immuno-oncology is encompassed by Chimeric Antigen Receptor T cells, or CAR T cells. So many different approaches are taken to recognize what was ultimately a similar product, which is the delivery of an artificial T cell receptor into a patient's own T cells. And so typically this involves withdrawing a sample of a patient's own blood, or in some cases blood from another donor, isolating and purifying the cells of interest, and then reprogramming them, typically with a virus, and most commonly with a virus that will introduce a chimeric antigen receptor that recognizes the CD19 antigen expressed on B cells, including B cell cancers. And so once this population of T cells is reprogrammed to include the new artificial T cell receptor, these cells can be grown up and reintroduced into a patient, typically after they have been given a preconditioning chemotherapy to wound their cancer cells, making them susceptible to attack by the CAR T cells. So this field has quite a bit of diversity in the approaches that are taken to create a chimeric antigen receptor T cell. We're now a few generations into what this means and what the technology looks like. But typically it involves a single chain variable fragment that recognizes the antigen, most commonly a CD19 on a target B cell, and then intracellular components which provide signals to our T cell. So this entire, because in this case, the cell is the drug, we need cell-based assays for the entire process of interrogating the performance and development and efficacy of this drug, which is a cell. And so throughout the workflow, whether it's in cell isolation and activation, engineering, growing up those cells prior to reintroduction, or cleaning them up and purifying, or ultimately characterizing them with a final QC, all of these things involve cell-based assays such as flow cytometry. And so cytometry is a critical component in this field. We can start at the beginning, where we are producing our lentiviral vector to introduce our chimeric antigen receptor to our target cells. Typically, this type of transduction has its success measured by the expression of a fusion protein, often a green fluorescent protein. And so we can detect this with flow cytometry and measure the success of incorporation of our protein of interest by the presence of that fluorescent protein in cells. But we can also look upstream of the production of that protein and look for other things in our cells, such as the introduction of messenger RNA that is not native to those cells. So we can use a technology called prime flow that enables detection of viral messenger RNA with flow cytometry in our cells of interest, rather than the expression of a fusion protein. So in this case, we see an example of the prime flow assay on a sample of cells where we're detecting both wild type and codon-altered viruses. And we see on the top left, we're able to specifically identify our wild type virus via prime flow and ElectroFluor 647. Top right, we can specifically identify the presence of RNA from our codon-altered virus. And on the bottom, we can see that we can mix and match those two conditions and identify which cells, not only which cells, but quantitatively, how much of that messenger RNA is present to our cells of interest. And this is often before protein has even been produced. So this can be useful upstream for analysis of introduction of CAR Ts. Once our cells have been reprogrammed, we grow up the CAR T cells. We proliferate them so that we have billions of cells to reintroduce into the patient. This is often monitored using a technique called cell proliferation analysis by dye dilution. This involves labeling a population of cells with a very bright dye that covalently labels those cells throughout their cytosol in a way that doesn't damage the cells, but allows us to track them over time. And as we see, our first generation indicated here corresponds to a very bright peak on our histogram. Every time those T cells divide, half of the dye migrates to each daughter cell. So our second generation of cells provides a peak that is half as bright as the parent. So as our cells continue proliferating, we see this ladder effect with each peak corresponding to one generation of cells. And so in a flow cytometry experiment with T cells, we can look, in this case, at CD3 positive cells isolated from PBMCs from a healthy human donor, in this case labeled with the dye cell trace violet, and incubated for 7 days. And we see one population of cells. These cells are not growing. We take the same cells and stimulate them, encourage them to proliferate with an antibody against CD3 and additional interleukin-2. What we see after 7 days is this striking histogram where each peak on this histogram corresponds to one generation of cells, where on the far right we have a peak corresponding to our parent generation, and then each peak to the left indicates an additional generation through which those cells have progressed. So we have a very clear measurement of not only that our cells are proliferating, but in this case, some cells are growing much faster than others. And because we can multiplex with flow cytometry, we can begin to interrogate what's going on in the early generations versus, or those cells that proliferate rapidly versus those cells that did not. And so this is very commonly used now in immunology techniques to characterize differential effects on T-cell proliferation. How different treatments over time give us variable proliferation. And so, as I mentioned, because we can multiplex, we can, for example, look at the phenomenon of activation-induced cell death, where these cells are growing. They're being given too much activation signal, and we're seeing cell death. So in this experiment, on the top left, we see cells, PBMCs, labeled with the dye cell trace far red. And in these plots, we're displaying CD3 positive T cells. So we see different peaks here corresponding to different generations of cells. Top right, we've also labeled these cells with cell event caspase 3,7 green apoptosis indicator. On this top right plot, we see a left-hand peak indicating our live cells and a bright peak on the right indicating apoptotic cells. So on the bottom, indicated by the blue arrow, we see a population of cells that has gone through many generations. They have had quite significant proliferation. They're also experiencing a dramatic induction of apoptosis, potentially as a result of overactivation. So it's through this multiplexing that we're able to really pin down what the effect of varying treatments is on our cells. So a few different things to look at as far as how we are characterizing our cells. One additional thing to look at is using flow cytometry to monitor the presence of our chimeric antigen receptor T cells in the peripheral blood, say in the case of animal models for research, where we'd like to monitor not only the immune response of those CAR T cells post-infusion, but also to determine the percent of T cells in that animal that are CAR T cells. And so we can see in two different patients or animals here that we see a dramatic increase in the presence of CAR T cells on the top row beginning in day 6, as we see cells begin to appear within that gate at day 10 and day 21. On the top row, we see a very high abundance of CAR T cells, and after on the final plot there on the right at day 153, we see our CAR T cells have diminished. And with second row, we see a little bit different pattern. But again, it's very important to track that our cells are still present over time in our model. So we've looked at 3 different approaches in detail at where flow cytometry is critical to understanding and discovering and developing new technologies for immunotherapy. So I hope as a result of this discussion, you have a better understanding of what immuno-oncology means and all of the different mechanisms it encompasses, as well as ways that we can use technologies available to us in a better way to provide these powerful analyses required to create this next generation of therapeutics. So Thermo Fisher Scientific as a company, as I mentioned, is poised to address it's our goal to provide a complete solution to help you in the process of developing these new technologies. And so at every step along the way, we're prepared to help you improve your processes and capabilities to drive your research to achieve your next answer. So I'd like to thank you all for your attention today, and I'm looking forward to answering your questions after this webinar. So thank you.